Click the bell icon and subscribe to our channel so that you will not miss any classes. Hello, welcome to class. As we discussed in the last class, the restriction enzymes, the discovery of restriction enzymes is helpful in constructing a recombinant DNA. And we have discussed that the restriction enzymes are of two types, restriction exonucleases and restriction endonucleases. Among these two, restriction endonucleases, which are capable of creating a pieces of DNA by cutting the DNA at a specific sites, is widely used, or it is used in reconstructing the recombinant DNA. And let us explore more things or more information about restriction endonucleases. There are 900 different types of restriction endonucleases have been isolated from 230 types of bacteria. Which means single bacteria is capable of producing more than one type of restriction endonuclease. And among the 900 different types of restriction endonucleases, only 30 of them are used in recombinant DNA technology. And the first restriction endonuclease isolated was HIM2, and it is from the bacteria Haemophilus influenza. From Haemophilus influenza, for the first time there was an isolation of HIM2 restriction endonuclease. It is first endonuclease. And there is another restriction endonuclease called EcoR1, and it was isolated by Hamilton Smith, and this is widely used restriction endonuclease. Among many, the widely used restriction endonuclease is EcoR1, and it is from bacteria Escherichia coli. And if you observe here, the naming of restriction endonucleases is quite different. Here, the naming of restriction enzyme is done. Naming of restriction endonucleases. It is very important. The naming of restriction endonucleases is done by giving 3-letter codes. 3-letter code will be given to restriction endonuclease. In this 3-letter code, the first letter in restriction endonuclease indicates the genus name of bacteria, from which the enzyme is isolated. We have discussed already, restriction endonucleases are produced by bacteria as a defense structures. So, from which bacteria we are isolating the restriction endonuclease, that genus name will be given to the restriction endonuclease. And the second letter indicates the species name of bacteria from which the restriction endonuclease is isolated. And the third name The third letter indicates the strain name. Let us take example so that we can understand it better. For example, I take hint 2. So here, for example, in hint 2, there are 3 letters. The first is H, the second letter will be IND, and the third letter will be 2, Roman letter. And remember, always the third letter is given by Roman letters. This is also important, Roman letters. And that Roman letter always indicates the strain name of the bacteria. So here, the H is first letter, and I told it is the name of genus. So here, the genus name is Haemophilus. And IND is the second letter, and it indicates the species name, that is influenza. And the third is Roman letter, which is a strain. In each species, depending upon the genomic differences, they will give different strain names. So here 2 is the strain name. And one more example, let us take EcoR1. So here in EcoR1, there are 3 letters. First is E, and it is always indicated by capital letter because it is a genus name. And the second will be CO, which is a species name, and R1 is the strain name. So here E indicates Escherichia and CO indicates coli, and R1 is the strain. So this is how the naming will be given to the restriction endonucleases. To take few more examples, ALU1, that is Arthrobacter lutis strain 1. Hint 3, Haemophilus influenza strain 3, same here. Hint 2, the strain is 2 here, the strain is 3 here. And BAM H1, here Bacillus amyloliquefaciens H1 is the strain. So likewise, many restriction endonucleases are there. We shall take up by discussing the mechanism with the examples. So here it's about the types of restriction endonucleases and the naming of restriction endonucleases. For theory exams, it may be asked for 2 or 3 marks. Explain the naming of restriction endonucleases. So likewise they may ask. Or for competitive exams like NEET CET, they may ask these. If hint 2 is the name of restriction endonuclease, then H indicates what? IND indicates what? 2 indicates what? So likewise, the question may be asked. So this is about the naming and the types of restriction endonucleases. And let us discuss the mechanism of action of restriction endonuclease. That means how it cuts the DNA in the next class.